In this video, we're going to cover the last few topics in Chapter 13, which include the polymerase chain reaction and DNA sequencing. When it comes to the polymerase chain reaction, it is also known for its abbreviation, which is PCR. Understand that this is an analytical technique where a fragment of DNA is amplified in vitro. In vitro means outside the cell. In order for a PCR reaction to work, we have several components. We need a DNA sample, which includes the DNA of interest. We're going to have primers, and these primers are going to be DNA based. We need a primer in the forward direction, and we need a primer in the reverse direction. We need deoxynucleotides. Taq polymerase, it is a specific DNA polymerase that works under different temperature conditions. We're going to have a buffer, which is going to include the important divalent cations like magnesium. And the container where the whole reaction is placed is called a PCR tube. Whenever we're performing a PCR reaction, we are going to place it in a thermal cycle. And a thermal cycler is the instrument for PCR. When it comes to the process of PCR, here on the right side of the figure, we have the three different stages that are occurring in the PCR process, and it's just illustrating one cycle. Understand that whenever we're running a PCR reaction, these steps are going to be repeated anywhere between 20 to 35 times. So, understand that the first step in the PCR process is going to be the denaturing step. In the denaturing step, as you can see, we're going to be heating up the reaction to 95 degrees. And at this point, that denaturing step is going to be separating the double-stranded DNA. Then in the annealing step, we're going to be cooling the reaction to around 55 degrees. This is on average. And understand that during this step, we're going to have those DNA primers bind the template. So you can see here, we have the reverse primer, and here we have the forward primer. On the last part of PCR, we have the extension step. In the extension step, as you can see, we are going to be increasing the temperature to 72 degrees, and this is when the Taq polymerase is going to be synthesizing the new strand of DNA. Now, understand that during PCR, this process is going to be similar to DNA replication. But understand that in PCR, we have no replication fork. The new DNA strands are made similar to what we discussed before in the 5' to 3' direction. And because this whole process is timed, this takes less time than the process of replication inside the cell. Understand that there's many applications for PCR reaction. They include molecular identification. One of the things that it is discussed in the textbook is the whole idea of DNA fingerprinting. Understand that in DNA fingerprinting, what we're doing is identifying certain segments of DNA. We can also utilize PCR for drug discovery and even for the detection of pathogens. When it comes to genetic engineering, understand that the PCR process can be utilized in site-directed mutagenesis. And for site-directed mutagenesis, this is a technique that is utilized in the laboratory to create, for example, an enzyme that is not active because you're changing its DNA content, and that results in changes in the protein. We can also utilize PCR for gene expression studies, and this is the main type of reaction that is utilized for looking at sequencing, which that's what's determined the primary structure for a piece of DNA. So in DNA and sequencing, understand that this is based on the Sanger method. And in the Sanger method, understand that what we're going to be determining is the primary structure of the DNA. So in here, what you have is that in steps 1 through 6 is where we are performing a PCR reaction. And in this PCR reaction, as you can see, similar to the steps that were outlined before, we're going to be denaturing double-stranded DNA using heat. So that's the process of denaturation that we saw previously. Then we are going to be making multiple copies of the segments that we're interested in. We're going to be attaching a primer. Remember, this primer is also going to be DNA based. Then understand that you're going to be making 4 different polymerase solutions, and these polymerase solutions, as you can see, are going to be DD-NTPs, or in other words, dideoxynucleotides. The incorporation of dideoxynucleotides along with DNTPs, which are deoxynucleotides, what are going to be doing is that in this PCR reaction, we're going to be growing complementary chains until the termination die, and this termination die is going to be those DDNTPs, are incorporated into the chains. Then you're going to proceed to denature these growing chains, then through gel electrophoresis, we are going to be separating the solutions that were made with each of the individuals DDNTPs, and we're going to see the bands. Then after that, what is generated is going to be a map of the different sequences that we have of our DNA. So one of the interesting things about the Sanger method is that it is utilizing dideoxynucleotides. And the important thing to remember about dideoxynucleotides is that now at the 3' position, we are not going to have a hydroxyl group. So what this allows is for termination of the synthesis in the strand. So that's why when we're utilizing this method, we are stopping into smaller fragments, and this is what gives us those primary structures where we can determine what are going to be the sequences in the DNAs that we're trying to study.